Hello everyone, welcome to BioLearn with Harshani YouTube channel. Every second, millions of cells in your body divide to replace old or damaged cells. But how does this happen without making mistakes? Let's discover this amazing process known as the cell cycle. So today I will explain about cell cycle and cell division. The cell cycle means a series of events through which a cell grows, duplicates its DNA, and divides into two identical daughter cells. It is essential for growth, tissue repair, and reproduction. Have you ever wondered how a tiny baby grows into an adult with trillions of cells? Or how a small cut on your skin heals within days? The answer lies in one of the most amazing biological processes, that is cell division. In simply, cell division means one parent cell is divided to produce new cells. There are two main types of cell division, mitosis and meiosis. Mitosis means it produces two genetically identical daughter cells for growth and repair. Meiosis means which produces two genetically different daughter cells, each containing half number of chromosomes for sexual reproduction. By the end of this video, you will understand the difference between mitosis and meiosis, why they are important and how they help maintain the life. Okay, when it comes to the cell cycle, it has two main stages, interphase and M phase. M phase means where the cell division takes place. Before a cell divides, it spends most of its life preparing. This stage is called interphase and it makes up about 90% of the entire cell cycle. So this interphase is divided into three stages, G1, S and G2. G1, that is the gap before the duplication. During G1 phase, cells grows in size, it produces proteins and organelles, and further cell carries out its normal functions. And during S phase, the cell replicates its DNA, and during duplication, each chromosome doubles to produce identical sister chromatids that will become visible when the chromosomes condense at the beginning of mitosis. So the two sister chromatids remain joined to each other at their centromeres. This replication of chromosome during S phase is crucial because genetic material must be copied exactly so that both daughter cells receive identical set of chromosomes. So this G2 is the interval between the chromosome duplication and the beginning of mitosis. During this time, cell may grow but less than during the G1 phase. It also synthesizes proteins that are essential to the subsequent steps of mitosis itself. Did you see that in the cell cycle there is another phase called G0 phase? It is also called resting or quiescent phase. We know that normal cell cycle starts from G1, S and then G2 and then finally leads to the cell division. However, some cells leave the active cell cycle usually from the G1 phase. When a cell is entered into the G0 state, that cell does not replicate its DNA and also does not prepare for the cell division. However, the cell is still alive and metabolically active by performing its normal functions. Some cells can return from G1 state to the normal cell cycle when they receive the right signals. Actually, that also depends on the cell types. So, highly specialized cells such as neurons may remain in G0 for a very long time. Before a cell divides, it needs to make sure that all the components required for the cell division are properly prepared. One important structure involved in this preparation is the centrosome. In this slide, we can see how the centrosome changes during G1, S and G2 phase. In animal cells, the centrosome contains a pair of centrioles surrounded by pericentriolar matrix from which microtubules radiate. During G1 stage, one centrosome is present near the nucleus. Then during the S stage, the centrosome replicates, producing two centrosomes that remain very close together. Finally, the two centrosomes remain in extremely close proximity during G2 phase before the mitosis. Unlike animal cells, plant cells do not have a typical centrosome containing centrioles. Instead, they use multiple microtubule organizing centers to organize their microtubule network and form that spindle during the cell division. Okay, once the cell finishes interphase, now the cell is ready to divide. This process breaks down into two main steps, mitosis and cytokinesis. Mitosis, where the nucleus divides, cytokinesis, where the rest of the cell and cytoplasm divides into new daughter cells. To make mitosis easy to master, it will break down its key stages step by step. Prophase, prometaphase, metaphase, anaphase, and telophase. Let's dive right in. Let's look at the main stages of mitosis and cytokinesis, visualized step by step. First stage is the prophase. During this phase, chromosomes are condensed and become visible. Other thing is, centrosomes replicated during interphase move apart toward the opposite poles and then they begin to generate new microtubules. Other feature is nucleus started to break down and disappear. So these are the main things that happen during this prophase stage. Next phase is prometaphase. During this phase, nuclear envelope is started to break down. Therefore, it allows microtubules originate from centrosomes to invade the nucleus. So, chromosomes are attached with these microtubules through kinetochore structure. Here the kinetochore is a structure in the centromere region of each chromatid that is specialized for convergence. Here we can see there are three different types of microtubules. One type is astral microtubules, which are extended out from centrosome toward the cell's periphery. Other one is the kinetochore microtubules, which are attached with the kinetochore structure of a chromatid. Third one is the polar microtubules, which are directed towards the middle of the cell. Next phase is the metaphase. Here we can see all the chromosomes are lined up on the metaphase plate and then these sister chromatids are facing up to the opposite poles. When it comes to the anaphase, sister chromatids are separated and they are moved to the opposite poles. Final stage is the telophase. During this phase, again nuclear envelope and nuclei are reformed. Another thing is the spindle fibers are also started to disappear. Here chromosomes become uncoiled and DNA appears as the chromatin fibers in this stage. Finally, during the cytokinesis, the cytoplasm is divided and form two daughter cells with identical nuclei. That means at the end of this process, actually one parent cell can form two identical daughter cells. Let's look at how the cytokinesis is occurred in both plant cells and animal cells. Because animal and plant cells have fundamental structural differences, they achieve cytokinesis in two completely different ways. When it comes to the animal cells, after chromosome separation during mitosis, the cell marks its midpoint and builds the actin-myosin contractile ring, which is anchored to the inner surface of the cell membrane. When this ring contracts, membrane is pulled inward, cleavage furrow. This furrow deepens, creating continually until the opposite cell membranes meet in the center. Then, When these membranes fuse, they break apart and the cytoplasm is divided, and finally it forms two daughter cells. Actually, what it happens in the plant cells, because of rigid plant cell wall in cells, they can't pinch inward. Instead, vesicles fused from the Golgi apparatus form the cell plate along the equator. Then it builds a new cell wall from the inside out. Finally, it creates two daughter cells. This is how the cytokinesis is happen in both animal and plant cells. Now, how does the cell make sure everything goes smoothly without rushing or making mistakes? That's where the cell cycle checkpoints come in. We can think that these checkpoints as quality control step signs during the cell's life cycle. At key stages, the cell pauses to ask crucial questions. We know that during interphase, cell is grown in size and proteins are synthesized. Before it enters to the S phase, then some questions are asked as checkpoints. Is cell of sufficient size? Have proper signals been received? If the answers are yes, then it will go with the duplication of chromosomes and centrosomes. But the answer is no, it will not go with the S phase and until all requirements are fulfilled. After the chromosome and centrosome duplication, before it moves into the mitosis process, then it will ask some questions. Have the chromosomes been completely duplicated? If the answer is yes, then it will enter to the mitosis process. But if the answer is no, it will not go with the mitosis process until all requirements are fulfilled. After the metaphase stage, then some questions are asked as checkpoints. Have all chromosomes arrived and aligned at the metaphase plate? If the answer is yes, then it will initiate the anaphase stage by separating the chromosomes to opposite poles and finally undergoes with the telophase and cytokinesis. But if the answer is no, it will not go with the anaphase stage until all requirements are fulfilled. Likewise, these checkpoints guarantee that the cell only divides when it is completely ready, ensuring the accurate duplication and separation of chromosomes. Now we have covered about mitosis. Let's turn our attention to meiosis. Meiosis is a specialized type of cell division designed to produce gametes like sperm and egg cells. Unlike mitosis, which maintains the chromosomes number, meiosis reduces it by half. Meiosis starts with a single diploid, that means 2N cell, then the DNA duplicates once and then it follows two consecutive cell divisions called as meiosis I and meiosis II. But there is no extra duplication between meiosis I and meiosis II. The ultimate result is one parent cell gives rise to 4 unique haploid daughter cells. Before a cell can even enter meiosis, it first has to get ready, and that preparing phase is interphase. Just like in mitosis, during interphase, the cell expands and grows. It completely replicates its DNA during S phase, and by the end of this preparation stage, every single chromosome consists of 2 identical sister chromatids attached at the centromere. With its genetic material fully duplicated and ready to go, the cell is officially prepared to enter meiosis I. So let's start with meiosis I and see exactly what happens to the chromosomes. First, we begin with Prophase I, which is the longest and one of the most important stages of meiosis. In Prophase I, chromosomes become visible and homologous chromosomes pair up. Here the crossing over occurs where the homologous chromosomes exchange DNA. In this stage, a nuclear envelope is also started to break down and spindle fibers are also formed. When it comes to the metaphase I, homologous chromosomes pairs are lined up at the center of the cell, and these spindle fibers are attached to each chromosome. During the anaphase I, homologous chromosomes are separated and they are moved to the opposite poles. However, the sister chromatids are remained attached through their centromeres. When the cell moves to the telophase I, then chromosomes reach to the opposite poles and nuclear envelopes are reformed. Then the cell divides into two haploid cells. Each daughter cell contains 1/2 number of chromosomes in the original parent nucleus, but each chromosome consists of two sister chromatids joined at their centromeres. Next, cytokinesis will happen. In most species, cytokinesis follows telophase I, with daughter nuclei becoming enclosed in separate daughter cells. Since the number of chromosomes is reduced in 1/2, this meiosis I is called as reductional division. And there is a short interphase before beginning of meiosis II. During this time, chromosomes usually decondensed, in which case they must be recondensed during the process of subsequent second mitotic division. Here, in some species, the chromosomes simply stay condensed. The important thing is there is no S phase during the interphase between meiosis I and meiosis II. That means chromosomes do not duplicate during this interphase. Brief interphase between these meiosis I and meiosis II is known as the interkinesis. When meiosis II is started, the starting phase is Prophase II. Under Prophase II, the chromosomes are condensed again, and the nuclear membranes are also disappeared and the new spindle fibers are formed. When it moves to the metaphase II, the chromosomes are lined up one by one at the equator. So the spindle fibers are attached to the sister chromatids. When it comes to the anaphase II, these sister chromatids are separated and they move to the opposite poles. Finally, during the telophase II, nuclear membranes are reformed around each of 4 daughter nuclei, and the cytokinesis taken place by separating each nucleus in a separate cell. Then it results 4 haploid cells. So at the end of meiosis II, each daughter cell has the same number of chromosomes as the parent cell present at the beginning of the cell division. For that reason, this meiosis II is known as the equational division. So this is how the meiosis I and meiosis II cell divisions take place. Okay, let's take a quick recap. We have learned about both mitosis and meiosis, so now let's put them side by side and see the main differences between these two types of cell divisions. Mitosis occurs in somatic cells and germline precursor cells. Haploid and diploid cells can undergo mitosis. That means both haploid and diploid cells can undergo with the mitosis. Meiosis occurs in germ cells as part of the sexual cycle, and it only happens in diploid cells. Mitosis has one round of division, but meiosis has two rounds of division called Meiosis I and Meiosis II. Mitosis is preceded by S phase. In meiosis, chromosomes duplicate prior to Meiosis I, but not before Meiosis II. In mitosis, homologous chromosomes do not pair. When it comes to the meiosis, during the prophase of Meiosis I, homologous chromosomes pair along their length. In mitosis, genetic exchange between homologous chromosomes is very rare. However, in meiosis, crossing over occurs between homologous chromosomes during prophase of Meiosis I. In mitosis, sister chromatid is attached to its spindle fibers from opposite poles during metaphase. In meiosis, homologous chromosomes attach to its spindle fibers from opposite poles during metaphase I. In mitosis, the centromeres of the sister chromatids separate at the beginning of anaphase. In meiosis, the centromeres of sister chromatids remain lightly attached during the Meiosis I. Mitosis produces two new daughter cells identical to each other and the original cells. So the mitosis is thus genetically conservative. Meiosis produces 4 haploid cells, 1 egg or all sperm, of which can become gametes. None of these is identical to each other or to the original cell because meiosis results in combinational change. Mitosis is mainly responsible for growth, repair and maintaining the cells. Meiosis produces cells involved in sexual reproduction. If you found this video is interesting, press the subscribe button, like the video and share. Leave your comments in comment section. See you until the next. Thank you for watching.